Hello students, Mrs. Klein's here with your next section of video notes over mitosis, also known as somatic cell division. So let's first talk about cells. Why aren't we made up of just one giant cell that carries out all of the important life processes to keep us alive? Well, we need smaller cells, and many of them, to allow for more efficient movement of nutrients and waste across our cell membrane. So remember, from cell transport, materials enter and leave the cell through the cell membrane. The bigger your cells, the harder it would be to deliver nutrients throughout all of the cell. So cells need to remain small so that materials can continue to move through them through active and passive transport relatively quickly. So the goal of the cell size is to have a high surface area to volume, meaning the volume inside is low compared to the amount of membrane that covers the volume or the cytoplasm. So in essence, smaller cells are more efficient than larger cells. We talked briefly earlier about the different phases of the cell cycle. First thing worth noting is that this is where a cell is born. This is also where a cell spends most of its time, in interphase. So, interphase of a cell's life cycle takes up most of its time and is broken down into three parts. The G1 or gap 1, which is where the cell grows and prepares for division. The synthesis or S phase, which is where DNA is replicated, and when DNA is replicated, it results in two identical copies of DNA. And the G2 phase is where the cell is making final preparations for cell division to take place. So after interphase, the cell goes into what we would call the mitotic phase. The mitotic phase is broken down into two parts. The first is mitosis. Mitosis is this piece right here. And we'll talk about this in more detail. But there are four parts to mitosis, which includes prophase, metaphase, anaphase, and telophase. In cytokinesis, which follows the mitosis phase and takes place when the cytoplasm divides as well. So at the end of cytokinesis, you've got two identical daughter cells that are produced, and then the cells that are just produced go back into the G1 phase of interphase. So just kind of the life cycle of the cell. It just keeps on going. That's the important thing now to kind of notice, is that there's multiple stages involved. Okay, as I stated earlier, there are two parts of what we would call the mitotic phase of the cell cycle. Mitosis is the first part, and that's when the nucleus divides. So inside the nucleus, we know that during interphase, specifically the S phase, DNA replicated to prepare for cell division. During mitosis, the nucleus will divide so that each newly developed nucleus gets a complete set of DNA. Cytokinesis is where the cell division process completes, and that is when the cytoplasm divides along with all of the cellular organelles, so each new daughter cell gets a little bit of everything. But they each get a nucleus that contains a full set of chromosomes. Mitosis is a form of asexual reproduction, and it occurs in diploid cells. Di means two. In cells, an N represents the number of chromosomes that are located in the cell's nucleus. So 2 N cells, think 2 means di. Diploid equals 2. These include our somatic cells. Another way to describe somatic cells would be your body cells. So any cell pretty much in a human that's not a reproductive cell, so not a sperm or egg cell, is going to be a body or somatic cell. Examples include skin cells, lung cells, liver cells, heart cells, brain cells. Those are all somatic cells. So we'll talk about the process in which our reproductive cells are made in our next section. But mitosis does not occur in haploid cells. Hap, think of half, meaning they have half the number of chromosomes. Again, this is a completely different process called meiosis that we'll talk about in an upcoming section of notes. So let's talk through some major events of mitosis. First, DNA replicates during interphase. Mitosis is where the nucleus divides. And at the end of cytokinesis, you've got two identical diploid daughter cells, meaning they're genetically identical to the parent cell, and they each have a full set of chromosomes. I've spent some time talking about haploid and diploid, and this will be explored again and reviewed again in the future. But I just wanted to give you a graphic to show you the significance of what these cells are. So diploid cells have double the chromosome number. So in the human example, our diploid cells all have 46 chromosomes, where our haploid cells will have 23. Our body cells, or somatic cells, which is where mitosis takes place, will be diploid. In meiosis, which is the next type of process we'll talk about after this section, occurs in our reproductive cells or gamete cells, so sperm and egg cells. The goal of mitosis is to ensure that all of the daughter cells that are produced have the same number of chromosomes and are genetically identical to the parent cells. So it just kind of ensures that it's like a duplicate copy of the original cell. Meiosis is going to be a little different and results in reproductive cells that are genetically unique. But kind of going back to where did that first diploid cell come from that makes up our body cells? So a diploid cell, called a zygote, formed during fertilization. Essentially, a zygote is a fertilized egg cell. So a haploid sperm cell and a haploid egg cell fused to form the first diploid cell with a full set of chromosomes. This is called a zygote. A zygote then would enter the mitotic phase, where it grows and develops into an embryo, fetus, and then ultimately into a baby. Let's talk about the 4 stages of mitosis. We call these PMAT. PMAT stands for prophase, metaphase, anaphase, telophase. So prophase, important events that take place here are the long strands of DNA condense or bundle around proteins called histones. They coil and bundle up on each other to make these nice, neat little packages of genetic material called chromosomes. And really what you see in one chromosome are 2 identical copies of DNA. So remember, during interphase, the DNA was copied. You can see there's one copy here, there's another copy here. So in our chromosome, one copy, another copy. Each strand is called a chromatid, so sometimes you'll hear these referred to as sister chromatids. The structure that holds the chromatids together is called a centromere. During prophase, the nuclear membrane starts to break down. Remember, in a cell, the DNA is always contained in the nucleus. For the chromosomes to separate or be pulled apart during mitosis, you need to first break down the nucleus, and then we'll see the formation of structures called spindle fibers. Spindle fibers extend out from the centrioles. These are part of animal cells, and they're like these little protein microtubules that will extend out and eventually will attach to the chromosomes. The next phase is metaphase. This is where the chromosomes will neatly line up in the middle of the cell along its equator. Spindle fibers reach out from the centrioles and attach to the chromosomes at the centromere. So think M for middle. Next phase is anaphase. Anaphase is where those spindle fibers start to shorten, and in that process, they separate the sister chromatids from each other. So chromosomes separate when sister chromatids are pulled apart. So now we refer to these as chromosomes. They get pulled apart to opposite poles of the cell, so think A for away or apart. Telophase occurs next, where around the different poles where the chromosomes were pulled, a nucleus will form around those chromosomes. The chromosomes now can relax and unwind into less bundled state called chromatin. So chromatin again is made of DNA. It's just not as tightly coiled up as we see in a chromosome. This concludes mitosis or PMAT, where the nucleus divides. The final stage of the mitotic cycle is going to be cytokinesis. Cytokinesis is the last step of the mitotic phase. And what happens here, as I stated earlier, is the cytoplasm gets divided up between the 2 new daughter cells along with the organelles. The cell membrane pinches off to form 2 new cells, so they call this the cleavage furrow. So this cleavage furrow will continue to divide until the cells completely separate from each other. In a plant cell, a different structure is involved. It's called a cell plate, and that forms between the 2 nuclei that were just formed to create a cell wall that separates the 2 new daughter plant cells. So, what are the results of all this trouble? The end result is that now you've got 2 brand new genetically identical diploid daughter cells, meaning each cell has the same number of chromosomes as the parent cell. Remember, humans have 46 chromosomes or 23 pairs of chromosomes. The new cells restart the cell cycle, so they go back and start all over again at the first stage of interphase, specifically the G1 phase, where it's growing and developing. Remember, this process is really important for humans and other organisms, and that it's the process that allows for cells to grow. And when we get new cells, those cells differentiate and become different types of cells. And they are also important for the maintenance repair of an organism because our cells do get worn out over time. Also remember from the earlier stages of human development that the fertilized egg called the zygote goes through the process to create a baby by the time it's born. Okay, so let's briefly explore what regulates the cell cycle and how these cells differentiate and become different types of cells. It's important to remember that there are cycle checkpoints built into the cell cycle, checking for damage to the DNA or mutations in the DNA. If there's a mistake that's caught, the cycle checkpoint gets activated and actually programs the cell to die. They call this apoptosis or programmed cell death. It'll self-destruct. Not all cells in your body will divide at the same rate. Some cells need to last you a lifetime. For example, your central nervous system, which is made up of your brain and spinal cord, those cells will last you a lifetime. The lymph cells in your eyeball will last you a lifetime. Female gametes, oocytes, which are used to make egg cells, last a lifetime. Some of our cells turn over really quickly. In the lining, your small intestine, for example, you've got tissue, and every 2 to 4 days, there's new cells dividing in those places to replace the damaged cells because you know that's where digestion happens and lots of acidic materials from the stomach pass through. So some cells last for days and some cells need to last you a lifetime. So you've got to take good care of them. There are times whenever abnormal cell cycles are observed, and this occurs when cells do not respond to the cell cycle mechanisms that program to stop dividing or program it to go into apoptosis. We call this uncontrolled cellular division or cancer. Cancer cells crowd out and take resources from normal cells. I've always had students ask me, well, why is cancer dangerous? Well, your normal cells, this is kind of how they spend their life. Okay, right here. In a normal cell, it'll spend 120 minutes in interphase, 60 minutes in prophase, and you can see there are different stages of mitosis that take a period of time. Looking at cancer cells, interphase gets shortened tremendously. So these cells are dividing very, very rapidly, and in the process, they're not maturing appropriately so that the cells are fully functional. They're more like these immature cells that don't really have a specific function. So they start to kind of just create what we would call a tumor, or they crowd out healthy functioning cells so that they can no longer do their job. Cancer can be caused by a couple of different factors. This is really kind of complicated, but something that we know in the environment that has caused cancer, we call those things carcinogens. So they're identifying more and more carcinogens, and they're labeling those once they identify them to be something that can possibly cause cancer. Things that can be carcinogenic include viruses. There's some viruses that increase cancer rates. There's radiation exposure, chemical exposures. So examples of chemicals that have been known to be carcinogenic or cause cancer are like asbestos and nicotine. There are also genetic factors or mutations that occur in the gene sequences that regulate the cell cycle that can lead to higher incidence or rates of cancer too. In mitosis, cells will continue to grow and divide. Well, once those cells increase in number, they start to increase in specialization, meaning that cells end up taking on different roles or function within the body. The process by which cells start off as these stem cells, these cells without really an identity, and then they specialize into muscle, blood, nerve, liver cells. This process is called cellular differentiation, and it is controlled by things like enzymes, proteins, and temperature. So if any of these factors are altered, you can imagine how that could possibly affect differentiation of cells in a developing organism. Stem cells are cells that go on to differentiate and become specialized cells. Stem cells are used in a variety of clinical applications. We'll talk a bit more about that, but differentiation of stem cells is controlled by proteins. So if the proteins are damaged or their DNA code is mutated, you may not see that differentiation occurring. There's 2 different types of stem cells to familiarize you guys with. The first is embryonic stem cells. Embryonic stem cells are going to be stem cells that make up embryos, and they really have the potential to become any kind of cell in the body. As we know, a zygote is a fertilized egg cell that will divide through mitosis to become all of the different organs and structures that make up a human. So embryonic stem cells kind of have this unlimited potential, where adult stem cells have more limitations. There are different stem cells in adults that still exist that we can use for different clinical applications. You may have heard of people receiving bone marrow transplants or being bone marrow donors. There are stem cells that live in our bones that can differentiate to become different types of blood cells or other cells in the body. There's a lot of new research happening in cellular differentiation and stem cells, or maybe ways we can take cells and kind of reverse them and put them back into a stem cell state.